Welcome to Guacamole and Salsa. This week, we'll discuss the major components of guacamole and salsa, such as avocados, chilies, and onions, as well as some of the foods that we typically eat with them, such as tortillas. We'll also examine how we experience the taste and aroma of the food we eat. Let's start by talking about the major component of guacamole, the avocado. Where most fruits store their precious calories largely in the form of sugars and starches, the avocado instead stores the energy in the form of fats. In fact, the avocado contains up to 30% oil by mass, and much of this is in the form of unsaturated fats, which are healthier than saturated fats. In contrast, an apple contains about 10% sugar by mass and very little oil. Another unusual characteristic of avocados is how they ripen. Most fruits will ripen while still attached to the plant, but avocados will only ripen after being removed from the plant. Once they're removed from the plant, they'll ripen within about a week. It's always important to keep in mind the climate in which produce evolved, as that can help us determine under which conditions the produce will handle well, and those conditions in which the produce cannot tolerate. Avocados evolved in the tropical climate in Mexico and Central America. This means that they never evolved cellular mechanisms to handle cold temperatures because they're simply not regularly exposed to those sorts of temperatures in nature. As a result, the cellular machinery that the avocado uses to ripen will get permanently damaged if exposed to the cold temperatures of your fridge. Once you put an avocado in the fridge, it will stop ripening and won't ripen further, even if removed from the fridge. Now, we can take advantage of this by allowing our avocado to ripen and then putting it into the fridge, where it will then stay safe to eat for longer than if left at room temperature. So we've talked about some differences between avocados and apples, but now let's turn to some similarities. When an apple is cut open, the flesh starts to turn brown. Avocados undergo a similar process. Both of these processes are the result of oxygen from the air reacting with certain chemicals within the flesh of the fruit. Now, often in biology, a chemical reaction will occur quite slowly under normal conditions, and so the organism evolves to use an enzyme. Enzymes are biological catalysts that speed up the rate of the reaction. We can reduce the rate of reaction, the rate at which the flesh turns brown, in a few ways. Since the reaction requires oxygen, we can slow the reaction rate by reducing the amount of oxygen present using plastic wrap. Note that you should wrap a bowl of guacamole with the plastic wrap directly on the surface of the guacamole. With the plastic wrap straight across the top of the bowl, there will still be a significant amount of oxygen present in the air trapped between the plastic wrap and the surface of the guacamole. Now, most enzymes are only active at specific pH ranges. We can take advantage of this by using an acid, such as lemon juice or lime juice, to deactivate the enzymes and dramatically slow down the reaction rate. And like most chemical reactions, we can also reduce the reaction rate by reducing the temperature. So let's examine what exactly is occurring when the flesh of the fruit turns brown. Phenols in the fruit, such as chlorogenic acid, react with oxygen with the help of an enzyme called polyphenol oxidase to form a quinone product. Note that the change in structure is the formation of the double bonds between the oxygen atoms on the six-membered ring at the bottom of the structure, along with the loss of the two hydrogen atoms. As a side note here, anytime you see a chemical name ending with ase, A-S-E, as we see here in polyphenol oxidase, that means that the chemical is an enzyme and that enzyme splits the chemical named just before the ase ending. So polyphenol oxidase splits the oxygen molecule, the O2, into individual oxygen atoms. Those individual oxygen atoms then react with the hydrogen atoms, removing them from the chlorogenic acid and causing the formation of the double bond between the oxygen atoms that are already bound to that six-membered ring. The quinone product then undergoes a series of other reactions and eventually ends up as melanin with a structure shown in the next slide. Plant tissue can be damaged in a variety of ways. By chemical damage from microbes and by mechanical damage such as when insects or animals bite into the tissue or when we cut into the tissue with a knife or even if the fruit falls to the ground. However the damage occurs, the plant has an evolutionary pressure to evolve ways to avoid this damage. Most plant tissue responds by releasing phenols to attack the invader's enzymes and membranes. Many of these phenols taste bad to animals and thus deter animals from eating that portion of the plant, and these phenols are a large part of why we avoid eating many plants or portions of the plant. They simply don't taste good. But these phenols also act as chemical warfare agents against microbes by reducing the reproduction rate of many of the microbes. Naturally, of course, microbes evolve to produce defenses to these chemicals, and so plants and microbes are in a constant evolutionary arms race to develop more potent chemical agents and defenses to those agents. The browning we see is a result of those phenols and enzymes acting on the flesh of the fruit. That is, it's collateral damage that the fruit inflicts on itself. Now, humans also undergo a similar browning reaction involving melanin. When our skin is exposed to the ultraviolet light from the sun, it causes damage to our cells. In an attempt to reduce that damage, we evolved to produce the same chemical, melanin, in our skin cells when exposed to sunlight. The more sunlight we're exposed to, the more melanin our skin produces and the browner our skin gets. That pigment then reduces the amount of damaging light that penetrates deeper into our bodies. Now, even without any exposure to ultraviolet, we all have some melanin in our skins. How much we produce is largely a result of where on the planet our ancestors evolved. Humans are primates, and we evolved from apes in the savannas of Africa. Of course, on the open savanna, we were exposed to lots of sunlight, especially because we don't have dense fur covering most of our bodies like most other land mammals, and so we had lots of melanin present in our skins. Over time, as we migrated out of Africa into the Middle East, Southern Asia, and Southern Europe, our skins began to lighten. As we migrated even farther from the equator, temperatures got colder, we began wearing more clothing covering our skins, and the angle that sunlight hits the surface of the Earth also decreases the amount of sunlight. As a result, those of us with ancestors from these regions have even less of the melanin produced naturally in our skins. Now you might be wondering why we evolved to have less melanin in our skins if the melanin protects us somewhat from skin cancer. We also need vitamin D to remain healthy, and our skin makes vitamin D when exposed to light. So there's a balance between allowing enough light through to make enough vitamin D to remain healthy, and reducing the rate of skin cancer enough. As we migrated farther from the equator, those of us with slightly lighter skin tones tended to make more vitamin D and had a slightly higher chance of passing our genes onto our offspring. Again, we can limit the enzymatic browning by reducing the pH, that is, by adding acids, by reducing the temperature, or by reducing the amount of oxygen present. We can also use boiling, since the high temperature destroys the browning enzyme. Remember that meat undergoes changes as we cook it due to the denaturing, that is, the changing of shape of proteins. Enzymes are usually made up of proteins, and so increasing the temperature usually causes them to denature as well. This change in shape usually permanently removes their catalytic activity, so they aren't active even after lowering the temperature back to room temperature. Of course, some foods will also undergo changes when boiling that make them less palatable or less desirable in certain dishes. For example, apple slices will become significantly softer when heated, which is fine for a pie, but it's less desirable if you want to eat the slices by themselves. Another vitamin that we need to stay healthy, vitamin C, or ascorbic acid, is an antioxidant. What we mean when we say a chemical is an antioxidant is that it oxidizes, it reacts with oxygen in the air, more readily than other chemicals. In this way, as long as there is some of the antioxidant present, the other chemicals present are largely prevented from oxidizing. Furthermore, vitamin C is also an acid, so adding it to our food lowers the pH. So if we put vitamin C on our cut apple or avocado, we'll prevent the enzymatic browning through both the antioxidant properties and the lowering of the pH, tending to reduce the activity of the browning enzymes. Okay, we've talked about the main ingredient in guacamole, the avocado. So now let's turn to an important ingredient in salsa, the chili. Chilies originate from South America and were spread throughout the world after Europeans arrived in the Americas. Columbus called these fruits peppers due to their spicy tang similar to the spice obtained by grinding black peppercorns common in Europe at the time. However, the capsicum family of chilies from South America are not related to the black pepper plant which originated in India. This name of pepper has largely stuck, but I'll refer to them using their Aztec name of chili. The spiciness of chilies is due to a single chemical called capsaicin. Before discussing the capsaicin that makes them special, however, it's important to note that capsicums are a rich source of vitamin C as well as a variety of carotenoids. The carotenoids include beta-carotene, which was first discovered in carrots, hence the name of carotene, astaxanthin, and lutein. Our bodies use these carotenoids to make vitamin A, which is important in a number of processes in our bodies, including in our eyes. During World War II, the Allies improved their rate of downing German aircraft over Britain as a result of the increased use of radar. Not wanting the Germans to catch on that it was this important technology that led to their increased air defenses, they began a PR campaign linking the beta-carotene found in carrots to improved vision in the defending airmen and anti-aircraft gunners. To this day, many people believe that eating more carrots can improve your vision. Alas, unless you already suffer from a vitamin A deficiency, which is relatively rare in developed nations, consuming extra carrots won't improve your vision. Let's turn back to the capsaicin that makes chili so spicy. The capsaicin is produced almost exclusively in the placenta of the fruit, that portion in the center of the inner cavity which holds the seeds. This isn't to say that there is no capsaicin in the rest of the fruit, as the capsaicin can diffuse throughout the other portions of the fruit, but that it will be found in substantially higher concentrations in the placenta. Now let's look at the chemical structure of the capsaicin molecule shown on the bottom left. The main features I want to point out is the long tail to the right, that long chain of carbon atoms ending with the branched methyl groups, the CH3 groups. That long hydrocarbon chain, and by hydrocarbon I mean it's made up of mostly hydrogen and carbon atoms, that long hydrocarbon chain is nonpolar. Now why does polarity matter? Well, you probably know that oil and water don't mix, but other substances, like sugar and salt, can dissolve in water. Let's define a few terms. A solute is a substance that dissolves into another. The substance that does the dissolving is called the solvent. So when you dissolve salt into water, the water is the solvent and the solute is the salt. It turns out that like dissolves like. So a polar solute, like salt, will dissolve well in polar solvents like water. And nonpolar solutes, like oil, will dissolve well in nonpolar solvents like gasoline. But a nonpolar solute won't dissolve very much into a polar solvent, and vice versa with a polar solute and a nonpolar solvent. Now we'll get back to this again in a few slides. So what happens when this capsaicin enters our body? First, of course, it activates the heat and pain receptors in our mouths, or other portions of our skin exposed to the capsaicin. Once it gets into our bloodstream, it has a variety of other impacts. Chief among them is that it makes us feel hotter, triggering cooling mechanisms such as increased sweating and higher blood flow in our skins. The capsaicin can also increase, to an extent, the body's metabolic rate, leading to some usage in weight loss supplements. The increased blood flow can also be used to reduce muscle pain as well as for relief from sinus congestion. However, the combination of the stimulation of pain receptors and the oily nature of capsaicin means it also makes a good crowd control tool. As the capsaicin enters the eyes, sinuses, or other areas of the respiratory tract, it causes pain and inflammation. So whether the capsaicin has gotten onto you from the cooking process or via the use of pepper spray, it can be quite difficult to remove. It can stay on your hands and later be rubbed into your eyes or other sensitive body parts, leading to quite a painful surprise. As a result, anything exposed to the oils should be washed quite thoroughly with hot water and plenty of soap. Remember that the water alone won't be enough to remove the oil, since the oil is nonpolar and the water is polar. The soap molecule has one end that's nonpolar that sticks to the oil, and another end that is polar that is dissolved into the water. When you're cooking with chilies, you can control the amount of capsaicin present in your dish by removing the placenta and seeds as shown on the right. Alternately, you can leave them in if you prefer a spicier dish. But once you've consumed the capsaicin, how do you calm the nerves in your mouth? Again, because the oils don't dissolve well in water, a drink of water will only temporarily cool your mouth down. And it might simply serve to spread the oil around the inside of your mouth, making the resurgence of heat and pain worse after you drink. Instead, you're better off dissolving the capsaicin oil in a drink of milk or alcohol, or by letting a little sugar serve a function similar to soap and help the oil dissolve into water. I've found that a sugar packet of the size typically provided by restaurants for coffee drinkers to be reasonably effective at reducing the burning sensation. Now, not all chilies have the same concentration of capsaicin. This led to a scientist by the name of Wilbur Scoville to develop the Scoville Scale for rating the spiciness of a chili extract. Pure capsaicin has a value of roughly 16 million Scoville heat units. We have bred a wide variety of different chilies ranging from the barely spicy sweet bell pepper at under 100 units, through jalapenos at 2.5 to 5,000 units, up to the extremely spicy ghost pepper at about 1 million units. Consumer-grade pepper spray is 1 to 2 million units, while police-grade pepper spray is significantly stronger at between 2.5 and 5 million units. So what exactly does the Scoville scale mean? Scoville noticed that he could taste the capsaicin at incredibly low concentrations, even down to 1 drop in a million. A specific mass of the chili is ground up and the capsaicin is extracted using alcohol. A measured amount of that alcohol is then added to increasingly large amounts of water until the spicy taste is just barely perceptible. The dilution necessary to reach this point is then the value on the Scoville scale. Now let's turn our discussion to the foods we typically eat with guacamole and salsa. Milled corn foods like tortillas, tortilla chips, tamales, and so on. When we grind corn kernels, we end up with particles of a variety of sizes. Large particles, roughly 0.6 to 1.2 millimeters in diameter, are called grits and are used to make the dish of the same name traditionally made in the southern U.S. Medium-sized particles form cornmeal, which we make into a variety of bread-like dishes such as polenta, johnny cake, and cornbread. Very finely ground particles with sizes under 0.2 millimeters are called corn flour. The corn flour is typically combined with other ingredients to make a variety of baked and fried foods. Traditionally, in Central and South American cuisine, the corn would be soaked in water and ground into masa, a flexible dough that could then be used in a wide variety of different ways, some of which are shown on the next few slides. If the masa is spread into a thin sheet and then quickly cooked, we get a tortilla. If the masa is used to encase a filling, with the entirety often wrapped in the corn husk, we get a tamale. We can also deep fry the tortilla to make the tortilla chips we often eat with guacamole or salsa. Alternately, we can deep fry the masa itself to make corn chips. Now let's turn away from chilies to another pungent group of plants, the allium or onion family. Alliums consist of a variety of plants including onions, garlics, and leeks. Note that we eat either the bulb or the leaves of the plant. In the case of onions and garlic, we eat the bulb, which is a storage portion of the stem of the plant, and in the case of leeks, we eat the leaves of the plant. All of the alliums have a fairly strong pungent flavor and odor due to a variety of sulfur-containing compounds that the plants evolved to produce in order to deter animals from eating the plant. While this chemical defense works against most animals, humans have found a way around it. While we don't particularly enjoy eating raw alliums in large quantities, we found that heating causes a chemical reaction that turns the chemical defenses into a much more palatable products that have an almost meaty flavor to them. The plants in the allium family absorb sulfur from the soil and store it in the form of sulfur-containing compounds that are stored separately from an enzyme produced by the plant. Once the cell membrane is broken open by cutting or chewing, the enzyme is released from the storage vacuole and begins to break the sulfur-containing compounds down into simpler, more volatile chemicals. When I use the word volatile, I mean that it readily leaves the solid or liquid phase and enters the gas phase. That is, a large fraction of the molecules make it into the air, which we can then breathe in and or smell if the molecules react with the scent receptors in our noses. Note that garlic contains concentrations of these sulfur-containing compounds roughly 100 times higher than the other alliums, and so it is much more potent. This then affects how we use garlic in our dishes. We only use garlic as a spice, in which we use a limited amount of garlic in the dish, typically including just a clove or two of garlic in the entire dish. On the other hand, we use onions as a significant component of our dishes, often including an entire onion in the dish, this despite the fact that the onion is significantly larger than the garlic. So why do onions make us cry? Onions contain syn-propanethiol S-oxide, shown here on the right. This small compound is quite volatile, so it quickly reaches our eyes once we begin cutting into the onion. Once dissolved in the liquid on the surface of our eyes, it attacks our nerve endings, causing pain. Furthermore, it breaks down into hydrogen sulfide, H2S, which is the chemical responsible for the rotten egg odor, and also sulfur dioxide and sulfuric acid. Naturally, these products further irritate our nerve endings, leading our bodies to produce tears in an attempt to flush out these irritants. We can minimize the tears produced by reducing the amount of syn-propanethiol S-oxide that makes it to our eyes. Cooling the onion prior to cutting accomplishes this in two separate ways. First, by reducing the volatility so that fewer molecules make it into the air, and also by reducing the reaction rate between the enzymes and the sulfur compounds in the onion. Alternately, you could don personal protective equipment that provides a seal that prevents outside air from reaching your eyes, such as chemical goggles or a full-face gas mask. Alas, most of us probably don't have such protective gear readily available at home, and will have to be satisfied with cooling our onions before cutting them. We have also bred so-called sweet onions with roughly half the concentration of sulfur-containing compounds and that are thus a more mild type of onion. Now let's turn to discussing how we experience the food that we eat. That is, let's talk about flavor. Now, flavor has two components, taste and aroma. Our sense of taste comes from the taste receptors on our tongues. We have 5 types of taste receptors, and those receptors activate when specific types of molecules bind to the receptor. Salts taste salty, sugars taste sweet, acids taste sour, bases taste bitter, and amino acids, the building blocks of proteins, taste savory, which is also sometimes called umami. Fruits and vegetables tend to have a high concentration of sugar, typically 10 to 15% by weight. Remember that in photosynthesis, plants capture the energy in sunlight and store it in the form of a specific sugar, glucose. Now, remember from the last video that organisms tend to use sugars to store energy only for the short term. In animals, unused sugars are converted into fats, but in plants, unused sugars are usually converted into starches instead. Starches are very long chains of sugar molecules, but because these starch molecules are so large, they're too big to fit into the same taste receptors that the sugars bind to. This means that they don't taste sweet to us. They also don't bind to any of the other taste receptors, and so starches are largely tasteless. While a fruit is undergoing the ripening process, the starches are converted back into sugars to make the fruit more appealing for animals to eat. Also, the acid content drops, raising the pH and decreasing the sourness, which we perceive as a further increase in sweetness. Now I want you to pause the video to think about why fruits contain so much tasty sugar. What does the plant get out of storing the energy in the form of sugar instead of in the form of the more stable starches? Remember that like all organisms, plants evolved a variety of techniques to increase the survival of their species. Plants reproduce in a number of different ways, but the most common is to produce seeds. Those seeds need to be dispersed away from the parent plant in some way. Some plants evolved to use wind to disperse their seeds. Those seeds typically are grown with structures with high wind resistance, such as the white filaments attached to dandelion seeds. Other plants evolved to entice animals to eat the seeds along with a tasty fruit, and these animals then poop out those seeds later on, and that manure also acts as fertilizer for the seeds. So ripe fruits tend to contain lots of tasty sugar to entice animals to eat them, and relatively few chemical defenses that tend to decrease the appeal. Portions of the plant that the plant doesn't want to be eaten, things like the leaves, stems, roots, and seeds, tend to contain more of the bitter chemical defenses. Now, plants obviously don't have emotions, so they don't want anything. But if an animal eats a plant's roots, that plant is less likely to be able to reproduce and spread its genes. So I'm anthropomorphizing the plant a bit to use the word want as shorthand for the evolutionary pressure on the plant to encourage or discourage certain animal behaviors such as eating fruits or not eating leaves, roots, and stems. We also mentioned earlier the savory or umami taste. That taste is the result of amino acids binding to the savory receptors on our tongues. Since meat contains lots of proteins, which are made up of amino acids, they taste quite savory to us. But most plants don't contain much protein. Some plants, such as tomatoes, oranges, and seaweeds, do contain significant quantities of amino acids. Because we tend to use ingredients that complement the rest of the dish, we tend to use these plants as ingredients in meat dishes, and we tend not to use other fruits in such dishes. For example, we often put tomatoes or ketchup on a hamburger because tomatoes contain lots of the amino acids, whereas we don't usually put other fruits like apples on a burger. Likewise, we tend not to put high amino acid content foods like tomatoes into sweet dishes or dishes that are not savory because they don't tend to complement the flavors that we have. They don't have as much sweetness. In contrast, we do put things like apples, which have a higher sugar content, into these types of dishes. Another aspect to the experience of flavor is touch, sometimes called mouthfeel. Some foods have the quality of astringency. This is the dry, puckery, or rough feeling that your mouth gets after drinking a strong tea or red wine, or after you eat an unripe banana or a peach. The astringent quality is caused by phenols with between 3 and 5 carbon rings called tannins. A few such tannins are shown here. Now, we call this class of molecules tannins because they're used in the tanning process. That is, we use them to tan animal hides into leather. So why do these molecules cause our mouths to feel dry? Normally, your mouth feels wet because it's coated in saliva. But that saliva isn't just water. It contains enzymes that start to break down the food you eat, and it also contains other proteins that act as lubricant, making our mouths feel even slipperier than they would if saliva was pure water. But the tannins bind to the proteins in our saliva, causing them to clump together. This increases the friction between them, and we lose that well-lubricated feeling. This then makes our mouths feel dry, even though there is still plenty of water present. But why do plants produce these tannins in the first place? The tannins interfere with bacterial and fungal proteins, so plants evolved the ability to produce them to reduce the growth of these microbes. The fact that the tannins also bind to the proteins in our saliva to cause the astringent feeling is an unintended consequence, although it likely also helps the plants deter animals from eating those portions of the plant. Let's look at the case of the unripe banana. If an animal eats a ripe banana, the tiny seeds inside the fruit can get dispersed in the animal manure. But if the animal eats an unripe banana before the seeds are mature enough, then the likelihood that the seeds sprout after passing through the animal's digestive system goes down. So the banana plant has an evolutionary pressure to encourage animals to eat the banana fruit only once the seeds have developed enough to survive passage through the digestive tract. The banana plant responds by creating the tannins in the unripe banana to dissuade the animal from eating the unripe fruit, and then destroying the tannins in the ripening process to remove that negative experience from the ripe fruit. Animals then evolve and learn to avoid the green banana and wait until the fruit turns yellow before eating it. Now, a small amount of astringency can add a feeling of substantialness to a dish, but too much astringency can be quite annoying. Furthermore, the astringent sensation becomes stronger and stronger with each dose of tannins. This means that where most flavors become less prominent upon repeated exposure, that is, on each subsequent bite of the dish, the astringent feeling becomes more and more prominent because more and more of the proteins in your saliva have become bound by the tannins. Yet another sensation tied to flavor is that of pungency. This is the hot sensation of spices like chilies, black pepper, ginger, mustard, horseradish, onions, and garlic. Just like the tannins, the active ingredient in each of these cases is a chemical defense intended to repel animals or deter microbial growth. And each of these chemical defenses causes us to feel pain when consumed, although the amount of pain varies a little bit depending on which chemical we're eating. But why do we continue to eat these foods? Why do we enjoy this painful sensation? It turns out that eating spicy foods may be the flavor equivalent of riding a roller coaster. That is, our bodies give off signals warning us of danger in our environment, but because we know that the danger is well contained and that we're not really in danger, we can savor the feelings of vertigo, shock, and pain for their own sakes, without actually putting our lives in danger. Remember that we are animals and our bodies retain many of the processes animals evolved over millions of years. When we sense danger, our bodies release a variety of hormones that increase our metabolism, increase blood flow, and heighten our awareness. Suppose that you're living thousands of years ago on the African savanna and you happen upon a lion. Your body responds in such a way as to maximize your chances in the fight or flight situation that you find yourself in. You've increased blood flow, breathing rate, and so on in order to allow your muscles to best respond, whether that means you're fighting off the lion or you're running from it. Once you perceive yourself no longer in danger, that is, you've either killed or driven off the lion if you chose to fight it, or you've escaped from it if you chose to run, those hormones are removed from your body and new hormones are released to calm those processes down. This second group of hormones often makes us feel good, and there's a good evolutionary reason for them to do so. Whatever you did, whether you fought or you ran, whatever you did worked. That is, you're still alive, so your body rewards those decisions in an attempt to increase the likelihood that you'll make similar choices in the future, and thereby increase the likelihood of you passing on your genes to your offspring. If, on the other hand, you get injured during your encounter with the lion, you experience pain. This is also an evolutionary pressure to incentivize you to avoid acting in a similar way in the future. But too much pain can also distract you or cause you to go into shock. so your brain also releases pain-relieving chemicals to reduce the pain that you feel. So, turning back to when we eat spicy foods, we feel pain upon eating those pungent chemicals, so our brains release pain relievers. Then, once we remove the pungent chemical from our mouths, the pain recedes and we're left with a pleasant feeling from the pain relievers. Furthermore, the irritation of our mouth increases its sensitivity to other sensations. That is, we notice more and/or different aspects of other sensations involved in taste. So the spiciness may cause other sensations to be felt more strongly. But why do some people enjoy spicy foods while others avoid them? Our sensitivity to pungency tends to decrease with exposure to it and lasts for a few days. So those people who regularly eat pungent foods can tolerate spicier dishes than people who rarely eat pungent foods. But taste isn't the only sensation involved in the flavor experience. Aroma is also quite important. As we eat, volatile molecules leave the liquid or solid food and enter the air. They then travel up into our nasal passages to be detected by receptors in our noses. Some of these molecules enter our noses when we breathe in outside air, but many enter via a tube that connects the back of our mouths with our nasal passages. In this way, we still experience the aroma of our food when we're chewing, which also mixes up little bits of food with the air in our mouths. Where we only have 5 different taste receptors, we have hundreds of different aroma receptors, and each one binds to a very specific set of volatile compounds. The aromas they are used to typically are composed of a large number of different compounds in very specific ratios. Vegetables and spices are typically characterized by a dozen or so compounds, whereas fruits are often characterized by hundreds of different compounds. Of the many compounds that cause us to experience a particular aroma, usually a few are the major contributors, while the remainder supply a broad background of supporting, enriching notes. Many artificial aromas are produced by combining the major compounds that make up an aroma that we find naturally. This simpler mix is usually enough for us to identify the aroma, but we can also tell that something isn't quite right with the smell either. We can tell that the diverse background of other low-concentration compounds is missing. Now, normally when we're together in class, this is when I pass around a variety of different chemicals for students to smell. Obviously, I can't do that with a video, so unfortunately you're going to miss out on the smells of these chemicals. The first two, ethyl benzoate, shown on the top right, and butyl benzoate, are both esters. An ester is a type of hydrocarbon which has a carbon atom bound to 2 oxygen atoms, one of which is doubly bonded to the carbon. The ethyl part is then attached to the oxygen atom that has a single bond with the carbon atom. Ethyl means 2 carbon atoms, while butyl means 4. So the ethyl benzoate, shown here, has 2 carbon atoms to the right of the ester. One is explicitly shown in the CH3 group, and the other is implicitly shown at the intersection between the 2 lines between the O and the CH3. The butyl benzoate adds 2 more carbon atoms to this chain. Both of these esters have a fruity aroma, but you can't identify which fruit because they're present in many different fruits. It's the combination of many different chemicals similar to these two, and the exact ratio between those chemicals that allows you to differentiate between the aroma of, say, an apple from a pear. I'd also like to point out that your nose is able to differentiate these ethyl benzoate from butyl benzoate, even though their chemical structures are so similar. This is just one example of how our noses are really quite good chemical detectors. Carbon disulfide is the next structure shown, with 1 carbon atom and 2 double-bonded sulfur atoms. This has a rotten egg smell to it. This, like many other sulfur-containing compounds, has a strong, unpleasant odor. The negative reaction we have is an instinctual response to sulfur compounds, as there are places on the planet with high concentrations of sulfur compounds in the air, and breathing in those compounds can kill us. The next structure shown is trans-cinnamaldehyde. An aldehyde is a hydrocarbon with a structure similar to the ester, except that the singly bonded oxygen is replaced with a hydrogen atom. As the name suggests, this is one of the main components in the aroma of cinnamon. Another major component in this aroma is cinnamic acid, which has a very similar structure, except that the hydrogen at the far right is replaced with an OH group, turning the alcohol into a carboxylic acid. The last structure on this slide is that of 4-hydroxy-3-methoxy-benzaldehyde, better known as vanillin. As the name suggests, this is the major component in the aroma of vanilla. While the name may be daunting, it's made up of a number of smaller parts. Let's start at the right side of the name, since that's the most important part. The end of the name is aldehyde, So this must contain an aldehyde group like we see in the trans-cinnamaldehyde. Sure enough, we can see a similar group on the top right. The benz part denotes a benzene ring, which is the six-membered ring of carbon atoms in the center with the 3 double bonds. Note that we have similar benzene groups in both of the benzoates, the trans-cinnamaldehyde, as well as many of the compounds that I've shown structures for on other slides. The methoxy is a combination of a methyl group, methyl means one carbon atom, with an oxygen to form an ether, so that's the OCH3 group on the bottom. Hydroxy means an alcohol group, so that's the OH group on the left. Now, the numbers denote how many carbon atoms the hydroxy and methoxy groups are from the aldehyde, with the aldehyde starting at carbon number 1. So the methoxy group is on the 3rd carbon as we progress clockwise around the ring, while the hydroxy group is at the 4th carbon. The last 2 compounds are D-carvone and L-carvone, although sometimes we'll call those plus or minus carvone or S-carvone and R-carvone. These 2 compounds have the same chemical formula with all the atoms in almost the exact same way. The only difference is that they're mirror images of each other, similar to the way your hands are left and right hands are mirror images of each other. In these structures, all the lines representing the chemical bonds are parallel to the plane of the screen, except for the lines represented by the dashed lines and the triangular-shaped wedge. The dashed lines indicate a bond going away from our eyes, while the triangular wedge represents a bond coming towards our eyes. Note especially that the triangular wedge has the broader end towards our eyes, while the narrower end is away from our eyes. In this way, it acts like a perspective drawing of a rectangular bond in which the near end would appear larger than the far end. So in these 2 structures, the hydrogen atom is below or behind the other atoms, while the group on the bottom is above or in front of the atoms in the 6-membered ring. That is, imagine the hydrogen atom as deeper into your screen and the group at the bottom as closer to your eyes. The L-carvone smells like spearmint, while the D-carvone smells like caraway. If you're not familiar with caraway, in the US, it's most often used by adding the whole seed to rye bread, although the oil from the fruit is often also used in some soaps. Now, roughly 5% of the population cannot distinguish between the 2 smells, but most of us can. I am unfortunately in that 5% of the population. I can't tell the difference between these 2. But when I've had students smell them, most of my students can detect the difference between these 2. So I want to point out that this means that our noses are capable of differentiating between chemical structures in which the only difference is that they're mirror images of each other. So again, our noses are really quite sophisticated chemical detectors.